Good morning to all. Today we are going to discuss about molecular drive. Molecular drive is a term coined by Gabriel Dover in 1982. Molecular drive, that means it is an evolutionary process like natural selection and neutral drift, that genetic composition of a population through generation. Here, what is meant by molecular drive means it is a type of evolutionary process, for example, like natural selection or neutral drift, that is the genetic composition of a population through the different type of generations. It is distinct from natural selection and neutral drift in that it emerges from the activities of number of mechanisms of DNA turnover, that means MOT, such as gene conversion, unequal crossing over, slippage, transposition, retrotransposition, and so on. That means the natural selection and the neutral drift, that is distinct from each other, and that is emerges from the activities of number of ubiquitous mechanism of DNA turnover, such that gene conversion, unequal crossing over, slippage, transposition, retrotransposition. These all over the mechanisms of DNA turnover. Molecular drive can also act in bacteria where parasexual process such as natural transformation, that it can cause DNA turnover. Here, the molecular drive that it can also act in, or that is also present in bacteria where parasexual process such as natural transformation, that it can cause DNA turnover. Turnover and its evolutionary consequences occur only in genus that have several copies within same individual, so called as families of genes. That means turnover and its evolutionary process or consequences occur only in genes and that have a several copies within the same individual, that is called as the families of genes. Turnover is a consequence of a frequent molecular exchange between copies of a family. Okay, then what is meant by turnover means it is a type of a consequence of different frequent molecular exchanges between the different copies of the family. The concept has been proposed to extend to diversification of the multi-gene families. There are different multi-gene family, and the concept is to proposed to the extend diversification of the multi-gene families and the mechanisms of DNA turnover. Here, that means MOT. First mechanism is gene conversion. Then second one is unequal crossing over. Third one is the slippage replication, and fourth one is transposition. Fifth one is retrotransposition. Then gene conversion means it is a one DNA sequence replace a homologous sequence. Then second type of mechanism is called as the unequal crossing over. Then third type is the slippage replication. That means is one explanation for the origin and evolution of repetitive DNA sequence. The fourth one is the transposition. Fifth one is retrotransposition. And here the turnover much have potential to retard, maintain or accelerate the rate of DNA differentiation between the population. Okay, turnover means that is have the much potential to retard or maintain or accelerate the rate of the DNA differentiation between the population. Gene conversion is a process by which DNA sequence replaces a homologous sequence such that sequence becomes after conversion event. That means gene conversion is a process by which one DNA sequence replaces the homologous sequence. Okay, one DNA sequence that can replace a homologous sequence such that a sequences become after conversion event. Gene conversion can be either allelic, that means can be allelic or ectopic. Here means allelic, that is one allele of a same gene that can replace the another allele. Then second one is the ectopic, that means meaning that one paralogous DNA sequence converts the another. That means gene conversion that can be either allelic or ectopic. In the case of allelic, that means one allele of a same gene that can replace the another allele. And the second is the ectopic, that means one paralogous DNA sequence that can be converts into the another. Then sequences are paralogous if they were created by duplication event within the genome. That means the sequences are paralogous if they were created by duplication event within the genome. And here there is a one diagram explained by here red color, that is a gene, and that around that is small rectangle portion that is called as chromosome, and that single gene that is going for duplication, then the single gene become doubled, then that become two type of genes, and that gene duplication that's again goes for the diversification. Okay, then two type of gene duplication. The gene color is a red color in the time of gene duplication, and that gene duplication that become diversification, then the red color is turned into the blue color, and that is goes for the growth of the multi-gene family. Okay, that is gone to the different type of family, that is the several group family, that is the multi-gene family, and it's become several type of color, that is not for any color, that is represented for a multi-gene family. Okay, and the two they are different by species split, that is two rectangles for each side, that is it's a species split, then it is for homogenization. Okay, both are going for the homogenization. And on the next diagram, that means here there is a one male and there is a one female. As we all know, in the male we can know that it is sperm, and the female in that is egg. That means they are both together for the sexual fusion and it produce that offspring. Okay, here means an altered DNA sequence, that is the small triangle portion in we can see in the portion of the male, that is a small altered DNA sequence that is spread through a sexual population as a consequence of a sex combined with several mechanisms of DNA turnover, that is MOT, such as gene conversion and transposition. Okay, that means that in the case of male, then we can see that small triangle portion, that is an altered DNA sequence in the male and female. In the male, that is sperm, and from the female, that is the egg, and that is sperm and egg goes for the sexual fusion, and that produce two type of offspring. In the case of male, we know that in the case under the sperm, the first circle that is contained with one sperm and one egg. In the case of sperm, that is two altered DNA sequences, and the egg, that is one altered DNA sequences. In the case of female, that offspring that produces with two altered DNA sequences in both male and the female. And molecular drive can be considered as two-step process. The gradual spread of sequences variant through a genome, that means homogenization. Earlier we said by homogenization. And second one is through a population, that is called as fixation. Okay, molecular drive that can be considered as two-step process, that means gradual spread of a sequence variant, that is by two type through a genome, that means homogenization, and second one is through a population, that is called as fixation. Then the evidences for molecular drive. What is meant by the evidences for molecular drive? That means three lines of evidences. First one is the ubiquitous existence of MOT. Then second one is widespread observation of concreted evolution. The third one is the phenomenon of molecular co-evolution, where two or more genetic elements that functionally interact with each other, that have evolved in unison, that means or in community. Okay, there are three type of evidences for the molecular drive. The three lines, the first one is ubiquitous existence of MOT, then the second one is widespread observation of concreted evolution, therefore a widespread evolution observation, and the third one is phenomenon of the molecular co-evolution, that means where two or more genetic elements that functionally interact with each other, that involved in a community. Then in molecular co-evolution, interacting partners of a successful interaction between say A and B in one species and A dash with B dash in the another species, that have evolved together to extent that A can no longer interact with B dash nor can A dash with B. Okay, here means they are in molecular co-evolution, they are interacting partners partners of successful interaction, that means A and B in one species. Okay, one species is contained with A and B, and another species is contained with A dash and the B dash. In that means they together exchange. That means A can no longer interact with B dash, or nor can A dash with the B. This is just example for in the case of molecular co-evolution. Okay, this is a term of molecular drive. That means molecular drive, that term is coined by the Gabriel Dover in 1982. And what is meant by the molecular drive means it is a type of evolutionary process like natural selection and the neutral drift that can genetic composition of a population through the generations. And that means they are the distant from natural selection, the neutral drift, and that is by the activities of the mechanism of DNA turnover, such that gene conversion, unequal crossing over, slippage, transposition, retrotransposition. And there are different type of mechanisms of DNA turnover, that one is the gene conversion, second one is unequal crossing over, third one is the slippage replication, fourth one is the transposition, fifth one is the retrotransposition. And just we by saying that evidences for molecular drive, and that means three lines of evidences of molecular drive, then ubiquitous existence of a motive, then widespread observation of concreted evolution, then third one is the phenomenon of the molecular co-evolution. And there is just a small example for in molecular co-evolution. Okay, these all are the terms, or these all are the small topics in the case of molecular drive. Thank you.